So welcome to our biomarker talk today with Dr. Faisal Khan. Dr. Khan will give his talk. There'll be an opportunity to ask questions following, and please place your questions in the chat box and we'll read them out afterwards. So Dr. Faisal Khan is a professor at the University of Calgary. Dr. Khan is the co-founder and CEO of OncoHelix Inc, an organization that specializes in providing esoteric cancer genomic and immunologic testing. In his clinical role, Dr. Khan works as the director of the Hematology Translational Lab at the University of Calgary and as a clinical director of the Histocompatibility and Immunogenetic Lab in the Alberta Precision Labs. Dr. Khan obtained his Ph.D. in human molecular genetics in India in 2006. He completed his postdoctoral training and clinical fellowship at the University of Calgary. Dr. Khan is a certified diplomat at the American Board of Histocompatibility and Immunogenetics. And Dr. Khan has more than 21 years of experience in transplant immunology, cancer genomics, and histocompatibility and immunogenetics. Based on his research, Dr. Khan has published over 105 research articles in scientific journals like Science, Blood, Transplantation, and Blood Advances. His research has also been recognized by several awards, including the American Society of the Society of Hematology Achievement Awards in 2013, 2015, and 2018, and the American Society for Histocompatibility and Immunogenetics Scholar Most Clinically Relevant Study Awards in 2014, 2019, and 2022. Welcome, Dr. Khan.
Thank you very much, Brenda. I appreciate this generous introduction. Thank you very much, everyone, for joining for this talk. Brenda and I had a few discussions in the past where we talked about how this organization that I have co-founded together with some of the other faculty members at University of Calgary called OncoHelix has made a significant impact in understanding how cancer genomic profiling should be performed in this type of cancer. As many of you, I heard, have your loved ones who have faced or are facing this disease. The information that I'm going to share today may be very helpful for us to start thinking about what type of impact a genomic profiling can make, and for that impact, how important it is to ensure that the cancer genomic testing is performed with the right set of tools. Because if it's not performed with the right set of tools, then how bad an outcome could be. So I'll share my screen here. Good afternoon, everyone. And thank you, Brenda, once again for inviting me for giving this presentation here. What I would like to talk today is our experience in comprehensive genomic profiling for cholangiocarcinomas, and how that can help to guide a personalized approach for cancer treatment for this pretty bad disease. If I start with a brief introduction about this organization that's actively involved in this type of work called OncoHelix, then it's not an overstatement that OncoHelix is one of the leading programs in cancer genomics and immunology. The team overall who have created OncoHelix all comes out from the Faculty of Medicine at University of Calgary with over 350 publications. In this organization, we have tested over 1 million samples, over 750,000 NGS profiles, including at least 5,000 cancer profiles by now. It's by far, it is the most diverse CGP program in Canada. And as Brenda has mentioned, the team has achieved many awards, including American Society of Achievement Award, American Society of Histocompatibility Scholar Awards, and College of American Pathology Award, based on what the work that has been completed in last over 1 decade. And if we talk about the statement of this organization here, we have 2 arms, a translational research arm and a clinical diagnostic arm. So basically, it's a brain and barn model where we do active translational research, identify biomarkers, develop assays that can help in formal clinical testing and can be used for clinical diagnostics. And our goal, it's very straightforward, that no patient, specimen, or data need to leave Canada for the sake of a firm diagnosis and or a precise treatment plan. The inception of this organization came out of the fact that for a cancer genomic testing, majority of the specimens, tumor specimens, were going out to US, and patients or the pharma companies or the healthcare system was paying a very heavy price to get those type of testing done in US. Overall, we have 3 modules: molecular diagnostics, cellular and immunology, and transplant diagnostics. And for the sake of today's presentation, we will primarily be talking about molecular diagnostics. The genesis of this module, I would say, goes back to 1961, when Watson and Crick first time described the structure of DNA. And then we saw several discoveries and inventions which led us to where we are right now. And to mention a few, in 1973, the gentleman Kary Mullis invented a technique called polymerase chain reaction, which allows us to amplify a certain section of the DNA and make multiple copies of it to study it in a better way. And then in 1990, the Human Genome Project was launched with a goal to sequence the entire human genome and see what type of variations occurs between individuals. And then ultimately 2002, when the first draft of human genome sequence became available for us to use it as a reference sequence and build all future genomic programs around it. So just to mention, these 4 were very important time points in the scientific history of human genome. And among the plethora of informations that became available in 2002 after this first draft of human genome was declared, the most important one was this: that if we randomly select any 2 individuals, then these 2 individuals are 99.9% genetically similar. First, it was very disheartening that we are only 0.1% different from each other. But if you look at the total number of nucleotides in a human genome, then it's roughly 3 billion nucleotides, which means even 0.1% difference means there is a 3 million nucleotide difference between any 2 randomly selected individuals. And what these 3 million nucleotides can do, it can give us different phenotypes. It can give us different susceptibility to diseases. It can give us different immune response to the same infection. It can give us different action of a drug, and it could lead to a different treatment outcome. And again, for the sake of today's presentation, we will focus on the four: susceptibility to disease, immune response, action of drugs, and different treatment outcomes. Okay, so with that background, let's directly dive into this arm, that first module, which I initially said called molecular diagnostics, more in the context of cancer and specifically in the context of cholangiocarcinomas. So if you look at that module, it generally, in OncoHelix, it has three arms. The first arm is focused on molecular oncology testing. The second arm is focused on genetic disease detection. And the third arm is focused on residual disease monitoring. The molecular oncology testing is something which we primarily focus on in Canada, only for the same reason as I have explained before, that for the sake of cancer genomic profiling, samples were almost always being sent out to US and other programs to provide that comprehensive report. Very limited labs and very rarely in the public labs we see where these tests are funded or available. And that's why OncoHelix started in 2018 to build these capacity for the Canadian patients. And as you know, what this type of work can do, it helps in diagnosing the cancer correctly. It helps in providing a risk classification of cancer so that we do not diverge into try and see the result approach. Rather, we should be knowing that there is a poor risk associated with this cancer, and that cancer should be treated in a different way rather than a cancer which has a favorable risk profile. And finally, the genomic profiling identifies mutations that can be targeted by specific drugs. So the genomic profiling basically help in all these three streams, helping in diagnosis, helping in cancer risk classification, and identifying targeted therapies. And if we look at last 1 decade, that how this field has evolved from a research work to a bona fide clinical diagnostic investigation, then the central dogma is simple. Patient's tumor is collected, it goes into a black box, and a report comes out. Now, the shape, the color, the structure of this black box has evolved over last 10 years. It moved from simple nucleic acid extraction to polymerase chain reaction to more complex sequence library preparation. It moved from simple gel electrophoresis to next generation sequencing, leading to multi-facet data analysis using different bioinformatic tools, and then expert interpretation to ultimately get the final report. And all these changes have happened in last 1 decade. So if we look at those events, then it moved from single gene testing to multi-gene hotspot next generation sequencing profiling to comprehensive genomic profiling. And what does these three means? When we talk about single gene, it means investigating one gene for small nucleotide variants or small mutations or insertion deletion mutations that are present at a precise location in the gene in human genome. So investigating single gene for a certain mutation at one time. When it moved to multi-gene hotspots, that means simultaneously investigating multiple genes for different type of mutations, whether it could be nucleotide variants, insertion deletions, or copy number variations. And it also start including assessment of fusions, known fusions, when certain part of gene 1 fuses with some other part of gene 2, and the fused product, or called translocated product, has a lot of time cancer promoting activity as an oncogene. So the concept moved from single gene to hotspot multi-gene. And then more recently, it moves to comprehensive genomic profiling, where we not only do different type of mutations in multiple different genes, we also do known fusions as well as novel fusions, fusions which have not been reported before. And then more complex signatures, as you may have known, tumor mutational burden, which means the amount of mutation that is present in the tumor tissue compared to a normal sample. A high TMB may have better chance to respond to an immunotherapy, for example. Similarly, microsatellite instability, the markers which are extremely critical for so many colon cancers. So the work evolved from investigating one mutation in a gene to multi-gene to more comprehensive. And if you look at the impact of this evolution, then the first evidence comes from this cartoon. So 2011 study on non-small cell lung carcinoma. 2 single gene markers were tested. 30% of patients were positive for biomarkers, EGFR and KRAS. More than 60% have no mutation detected. A different non-cell small lung cancer carcinoma patient evaluated by hotspot multi-gene NGS. Now, 65% cases were positive for biomarkers and only 35% were negative. Why? Because now we are targeting more number of genes. Now we are targeting more number of mutations to evaluate this cancer. The very next study, 2017, in cancer discovery, where the samples were subjected to comprehensive genomic profiling. Same type of cancer, non-small cell lung carcinoma. End result, 85% cancer patients were found positive for at least one biomarker, and only 15% shows no mutation detected. So in the last slide we saw, as the technology moved from single gene to multi-gene to CGP, here we see the real impact that the numbers of biomarker detected moved from single gene to multi-gene to CGP increases significantly from 30% to 85%. Then comes the second evidence. If we will not be moving ahead that fast in these technologies, we will not be able to catch the progress in the new drug discoveries. If you see, again, non-small cell lung carcinoma patients, the FDA approved over 50% of biomarker-driven therapies have got the FDA approval between 2018 and I would say 2022, mainly because more and more advanced testings were available where multiple genes were evaluated, multiple variants were detected, which allows the discovery and approval of newer drugs that can target cancers. So we can clearly see that as we move from single gene to multi-gene to CGP, there are huge advantages, whether in terms of biomarker discovery or eligibility to biomarker-driven approved drugs. However, I want to say one thing, that although the technology has evolved, but we should not be simply ruling out single gene testing as something which is not good. It's still, single gene testing has its own advantages. In this slide, I have provided a comparison of a single gene testing versus comprehensive genomic profiling for us to understand where the merit is. If the need is high throughput, where we want to analyze hundreds and thousands of genomic region, then CGP. If the goal is to have a very highly sensitive assay that can detect mutation even at a rate of 0.5 to 1% using the cell-free DNA profiling, then the target is comprehensive genomic profiling. If the goal is discovery power, where you want to discover newer biomarkers and see how these biomarkers can impact the cancer cancer diagnosis, prognostication, or targeted therapy, then it's CGP. And it can work with majority of sources from FFPE tissues, so to circular tumor DNA to bone marrow, and with very, very limited amount of specimen quantity. However, if you are in a system where cost is a big issue, then single gene testing is better for one gene per target. If you're in a system where we do not have a huge bioinformatic infrastructure available for analysis, then the single gene testing analysis is relatively simple. If the goal is to have the turnaround time very quickly, then single gene is still the solution of that event. So there are advantages and disadvantages. And I would like to give you an example of other cancer called acute myeloid leukemia, which is a type of very aggressive blood cancer, very poor survival. And if AML patients have a mutation called FLT3, then patients can be treated directly with a drug called midostaurin. However, the midostaurin is most efficacious if it is given within 1 week of diagnosis. NGS profiling cannot be done within 1 week. So we came up with a strategy here that as soon as we get an AML patients, we perform a single gene test for FLT3 mutation reported within 5 days. If positive, patients get treated with midostaurin. And in next 2 weeks, the whole comprehensive genomic profiling of 50 genes become available for further risk classification and identification of other targets. So, CGP has huge advantages, but in many type of cancers, you need to be more pragmatic, where a combination of more than one approach is required. And I wanted to be very unbiased, though OncoHelix is a program which provides high-grade CGP, but still it does not mean that all the single gene testing and other multi-gene testing provided by other labs, including in our public healthcare system, are of no use. They are absolutely of great use. It's just as the field is evolving, we need to come up with a better strategies to make use of each of these approach in a proper way. Okay. Here is another example, a very, very nice study, 10,000 refractory cancer patients analyzed by comprehensive genomic profiling as well as by hotspot NGS panel. Like the result I showed earlier for the non-small cell lung carcinoma, this paper published in Nature Medicine in 2017 showed that 81% of variants were detected only in CGP profiling, and they were completely missed in hotspot NGS panel. In the next few slides, I will discuss why this is so critical. Okay. So with that background, what we have talked about, that how the field has evolved from single gene to multi-gene to CGP profiling, how it is impacting the biomarker discovery, targeted therapies, and what are the advantages of each of these approaches. So now we will be talking specifically what is more important for this presentation or this meeting is our experience of doing comprehensive genomic profiling for cholangiocarcinomas and OncoHelix. So this work was sponsored by Insight. As many of you know, that for some of their drugs, they were looking for FGFR2 fusions to be present. And until before they started working with OncoHelix, the samples were getting tested in different labs. and they were finding a frequency of around 1.5% patients being, or probably 1 to 2% patients being positive for FGFR2 fusion. 3 years ago, Insight made a contract with OncoHelix that OncoHelix will provide the comprehensive genomic profiling for cholangiocarcinoma patients, sponsored by Insight, mainly targeting FGFR2 fusion as a multi-center Canadian study. And as I've been saying, that OncoHelix has always been involved in having a very pragmatic approaches. We worked with Insight to come up with a nice solution that although a CGP takes 4 weeks, but we specifically analyzed FGFR2 fusions within 2 weeks and reported whether it is present or not to Insight, so that they can start enrolling those patients for their study for the drugs approved for by Insight. And then in next 2 weeks, we issued the full comprehensive report of 170 genes to the oncologist, that even for patients who are not FGFR2 fusion, but may be positive for other variants, the oncologist may still be able to treat those patients for those variants. So it allowed so much, such a nice win-win for everyone. Patient got these testing done free of cost. Insight got their information about FGFR2 fusions within 2 weeks, and OncoHelix was able to do a very nice study on this important cancer. So our first finding was all these mutations were detected in cholangiocarcinoma patients. Approximately 47% patients have targetable alterations in one way or other, class 1 or tier 1 or tier 2 classification. If we look a little bit more in detail, then IDH1 and FGFR2 fusions. I want to show here, guys, if you look at here, it's around 12% patients were positive in the Calgary data for FGFR2 fusion, compared to all other reports from other labs that were helping Insight before, where only up to 2% patients were deemed positive for this fusion. But this CGP was detecting up to 12% patients being positive for FGFR2 fusion. Other mutations consistent with other reports like IDH1, ERBB2, PIK3CA, and BRAF that were consistently detected. And I just put it together, some of the other studies which have shown these data, but all the other studies were retrospective. We were doing prospective, where every detection of a genomic alteration was used by the oncologist to plan the treatment and management of these patients, or were used by Insight to enroll the patients for their drugs. So this is, I would say, the meat, the slide is the meat of the whole presentation here. 245 patients, FGFR2 fusion, TST170 profiling. Okay. Based on the result that we saw in the previous slides, what we did here is we picked all the positive samples which were analyzed by this panel called OncoHelix 2, were reanalyzed by a smaller multi-gene NGS panel, which is a 52-gene panel. The rationale was that this panel is used by majority of the labs who are doing NGS profiling at this moment in Canada. And not in Canada, but I would say all over the world, because it is still an NGS profile, but a multi-gene, not a CGP profiling. And if we see vis-a-vis comparison of these two panels, then, as I said, 52 versus 170, there is a chemistry difference. It's called amplicon, it's called hybrid capture. I will not go in detail about it. But if you look at the rearrangement of fusion category, then both can detect fusions. However, focus panel can only do known fusions, which means this panel can detect fusion if both genes are present. So FGFR2 is one, and the partner gene, if it is present, it can detect it. versus the panel that OncoHelix was using. It's called TST170, called as OncoHelix 2 panel. It can also detect novel fusion, which means only one partner gene, if it's present in the fusion, the panel is able to detect the second gene without being present in the panel itself. It compares the sequence with the known human genome and provide the information that FGFR2 gene has fused with this particular gene, irrespective of the fact that whether the gene is present in the panel or not. And if you look at the end result, this is the data of 175 specimens, 92 variants, 16 CNVs, and 17 patients who were positive. So roughly, yes, I'm talking about 10% people having FGFR2 fusion. We found 27% of, sorry, this is a typo here. SNVs were missed by NGS assays, and the reason was mainly gene coverage. Focus panel was 52 genes. OncoHelix panel was 170 genes. But the most important result is here. 94% of cholangiocarcinoma patients were found negative for FGFR2 fusion when they were tested by the focus panel, compared to when they were tested by OncoHelix 2 panel. And the reason was 16 out of 17 individuals have novel fusion where FGFR2 was in rearrangement with a gene that was not part of the panel. So what does this mean? This means that a cancer patient thinking that he or she got profiled by NGS and is negative for a variant. If we are not sure whether the patient was profiled by the right panel, then that information means nothing. Example here, 94% of these patients who were detected FGFR2 fusion positive would have been reported negative if we would have been opting for a panel that's not focusing on novel fusions. And because of this, very critical and very nice result, Insight decided to continue with OncoHelix for another 2 years. We are still testing it for them for all over the country, and we now also have finalize an agreement with the C3 network where OncoHelix will be testing the cholangiocarcinoma patients, which are part of the C3 study for all over Canada, again with the same panel, based on this important finding that a CGP panel is probably the best way to detect these variants in these patients. So in the last few slides, I just want to show what type of panels are available with OncoHelix that can help in these type of profiling. We have 3 panels. OncoHelix 1 is a 324 gene panel, which does all these type of work, including TMB and MSI. A lot of you know this panel by the name of Foundation 1. In Canada, OncoHelix has clinically validated this panel offered by Roche. It's the same 324 gene panel, which is offered by Foundation 1, but here it is offered as OncoHelix 1. And the analysis done using the Foundation 1 software, exactly same panel through Roche Diagnostics. And I may say that at a much, much cheaper cost. At one stage, it was 1/3 of the cost what patients were paying when they were sending specimens to US. I don't know what's the current costing, so don't want to comment on it. But when we started, it was OncoHelix was offering this test at 1/3 of the cost of what is being offered in US. So I was showing OncoHelix 1, and then there's an OncoHelix 2 panel, the panel whose result we have seen so far. It's an Illumina panel where OncoHelix collaborates with an organization, Sophia Genetics, to build the bioinformatic pipelines and analyze these results. Extremely solid panel. We have seen very, very nice results. And then there's a third panel called OncoHelix CFDNA panel, which OncoHelix is bringing on board in collaboration with Memorial Sloan Kettering Access Program through Sophia Genetics. It's a 129 gene panel which does not require a FFPE biopsy. This panel can be done on blood samples by extracting cell-free DNA. Very critical for patients where biopsy is not possible. Very critical if you're trying to understand metastasis or measurable residual disease, whether the tumor is responding to a treatment or not. So each one of them have their own importance. OncoHelix 1 has the highest number of genes, and it can do tumor mutational burden and MSI. OncoHelix 2 is a very nice panel because it uses DNA and RNA, and it requires the limited amount of tumor cellularity. Only 10% tumor cells in a block have been validated to offer this test. And the cfDNA, where none of the biopsies are possible, still the test can be performed using cell-free DNA. And all major organizations, including Roche, Foundation One, Illumina, Sophia, and Memorial Sloan Kettering, are our partners in bringing these tests here for the Canadian patients. And if we talk about access and reach, then we have tried to have a full 9 yards. We work with hospitals, public payers, government programs that if they would like the testing to be performed, we offer these tests to these programs all over the world. Like right now, we are receiving samples from 14 different countries in Europe, Asia, and North America. We work for different clinical trials with national and regional reference labs. We work with pharma-sponsored programs, as the data I just showed recently for Insight. We do a lot of basic genomic research initiatives. We help in national registry collaborations. And if none of these is working, we also have a patient direct pay mechanism where if patient would like themselves to be tested, it is possible to get done through OncoHelix. And I would just summarize what we have discussed in the last half an hour or so, that CGP allows detection of a greater number of actionable genetic alterations, allows assessment of biomarker interactions. It helps from diagnosis to risk assessment to targeted therapies. It could ultimately lead to improved outcomes, and it allows better use of resources. Instead of doing 10 single genes in the same tissue with the same amount of DNA, you could do 324 genes. So there's a better use of resources in terms of tissue cost and expertise. And I would like to acknowledge all these people who, the organizations, University of Calgary, OncoHelix, Burjeel Holdings, which is our partner in Asia, based out of Abu Dhabi, all the funding agencies from Alberta Innovate, POET, CIHR, Alberta Cancer Foundation, who have helped in the research activities to build these assays and for these biomarker discoveries, and our partners in industry from pharma, like Insight, Illumina, Roche, Sophia, Connexia, which has allowed all us to be able to offer this type of services here to Canadians. I'll stop here and will be happy to take any questions. Thank you very much.
Thank you. There's one here. I think you already answered it though. Do you use tumor tissue or can you use blood samples for your?
Yes.
Yeah, you already answered that one.
I can. We can do both with these panels, depending on which panel we select.
And genetic testing versus CGP. How can you explain the difference of like the genetic testing? If somebody requires genetic testing based on if they have a biomarker for cholangiocarcinoma, should their kids or other family members get genetic testing? Like what % has to show up?
Right.
If that makes sense.
Right. Yeah, it does. In a very simplified language, Brenda, there are two types of mutations: the germline mutations and the somatic mutations. The germline mutations are the one which you inherit, which are there from the time of birth. Somatic variants, which occurs as we age, as we get exposed to different carcinogens, or several other reasons. So the tests which I mentioned here are all somatic variants where the variants are detected in the cancer tissues. However, if we look closely, then germline variants can only occur in two type of fractions, either 50% or 100%. 50% when they're heterozygous coming from one parent, 100% when they're homozygous coming from both parents. So in this test, although we do not detect germline or genetic mutations, but detect the somatic variants, however, if the variant allele frequency is anywhere above 35%, then we evaluate that variant in more detail and see if there are evidence of such variant being present as a germline variant in this cancer or in any other cancer. And in our report, we provide a comment that although this test cannot differentiate between a germline and a somatic variant, but there's a high likelihood that this variant is a germline variant and a germline testing is recommended. So the next step is do the germline testing for that variant. And if it turns out to be present, let's say in the blood sample or in a buccal swab, then the next step is the genetic counseling to see if kids or others need to be tested for it. So it goes in these three steps, Brenda.
I have one question here too. Is there value in retesting samples from deceased patients? I now realize, this is me, but I mean, I'm just reading it. I now realize that things may have been missed in the 2020 Foundation 1 test. This information could be valuable for my daughters as their mother and grandmother both had cholangiocarcinoma.
Yeah, so we could all, if the tissue block is available, the sample can always be sequenced to determine what's the cause of the sequence, what were the genomic profiling of those cancers. For example, so many studies that we are performing right now are all retrospective samples initiated by different researchers where the goal is to see if a mutation was present in the original biopsy and how it is linked with the overall responses or survival of the patient. So yes, it is possible. However, to get that, because the patient need to give the consent, and if the patient has not given the consent, the oncologist may not be able to recommend the testing. So it can be done only if it is part of a research study where a retrospective specimen can be evaluated. I'll try to find some information and let you know that you can contact. I think C3 could be an ideal platform, Brenda, that they are doing this type of studies, and if they can accommodate a retrospective specimen, whether the patient alive or not, we should be able to sequence it as long as the tissue is available. So I would strongly recommend to connect with C3, either anyone from Dr. Rebecca Hour's team, and they may be able to guide if this component can be a part of their study, and we should be able to sequence.
Any other questions? Well, I want to thank you very much for your time and sharing your expertise with us today. It was very, very informative. You're brilliant.
We're just trying to do a good job here, and honestly, this study is very close to my heart because the fact that the incidence of FGFR2 fusion detection is almost tenfold with what we are doing, make me believe that we are on the right path. And if we are able to provide these type of tests to as many patients as possible in the country, then why not? And we are not leaving any stone unturned, Brenda. We are talking with the public health program. So there are a few public health programs who have now contracted us to do some of these testing for their patients. funded by the provincial healthcare system. Pharma is absolutely sponsoring so much of these. A lot of credit goes to Insight for actually continuing to support this. And then also various research programs like C3. As I said, if nothing else work, even there's a patient pay mechanism that allows patients to spend very limited amount of money compared to what everyone was paying when we were sending the samples out to US. So
I do have one more question here, Faisal. Sorry, is there any value retesting as new tumors present over time?
yeah, absolutely. So tumors evolve over time. And they acquire new mutations, or some of the mutations which are present in a different variant allele frequency are now more evident. They are in a higher variant allele frequency. And also it all depends that which method was used originally when the testing was performed. Like if you ask me if the testing was done with a focus panel or that 52 gene panel or any any panel which was not doing CGP, then why not test it with the proper CGP panel now to have any additional information as possible, with the caveat that we may not be able to find anything additional, but if I have to do, if I had to make that call, I'll always try to do this.
Okay, that's good. Any other questions? Sorry, I thought we were finished and we're not, and that's great. You're sparking a lot of questions as we go along. Oh, one more. No, I don't see any more. That's it. All right. Well, thank you. This time I'm thanking you very much for your time. Really, really appreciated you sharing your expertise with us. It was very informative. And we're going to save this and put it on our website so that others can look at it.
Thank you very much for inviting me to share this data and this information with the team. And please feel free to reach out to us if you have any questions, if you need any further information. Thank you.